students, today we are going to begin with our new chapter, that is Structure of Chromosomes, Cell Cycle and Cell Division. This chapter is divided into two parts. Part A consists of the structure of chromosomes. Children, you have learned in the previous classes about the cell and its organelle. In the cell organelles, we have learned about the nucleus and centrosome too. So in this chapter, we are going to mainly focus on these two organelle and see how cell division occurs. So first we'll start with structure of chromosomes. What are chromosomes? Chromosomes are highly condensed coiled chromatin fibers. Now in your mind you have the question, what are chromatin fibers? Chromatin fibers are a network of very extremely long thin dark stain fibers which are found inside the nucleus. When we observe a cell under the microscope, we find that it is a non-dividing stage called interphase of the cell. When we observe the cell under the microscope, we will see its first stage, which is known as the interphase, which is also known as the resting stage. Children, you see I have marked here stages of cell division. Stages of cell division are mainly four types: prophase, metaphase, anaphase and telophase. The first which I have mentioned is interphase, that is the resting stage when the cell has to divide, when it plans to get divided. This we will learn in the second part of this chapter. So first we'll just focus on interphase and prophase. This is the structure of cell when it is in the interphase. You can see I have marked here the chromatin fibers and the nucleolus. As soon as it enters the first phase, that is the prophase, this chromatin fibers get condensed to form the chromosomes. Chromosome was first discovered by Walter Fleming in 1882. In the larvae of salamander. Salamander is an amphibian, and he observed that certain threads were found in its cell. Now let us see some glossary terms. When you will study about the chromosome further, you may find difficulty in understanding. So I am just explaining these parts. Chromatin network or thread, I have already told you what is it. These are highly dark stained long structures found inside the nucleus of the cell. Then there is chromatid. Chromatid are the chromosomes which are copied. You will find it when I'll show you the structure of the chromosome. Centromere, it is the point of attachment of the two chromatids. Centrosome. Centrosome you have learnt in the previous chapter, that is cell and its organelle, that it is an organelle which is responsible for the cell division. The centrosome is only found in the animal cell and it is very much important for the cell division. Next is chromosome. Chromosome is actually a DNA molecule which is packed into thread-like structures. So this was mainly about the glossary terms. When you will read the chapter further, you will get to know it. This is a typical structure of the chromosome. Each chromosome in its condensed form as visible during the start of the cell division consists of two chromatids. You can see there are two chromatids I have marked, and they are joined at some point about its length, which is called as centromere. This is the point of attachment of the two chromatids. This centromere function is, it serves to attach the spindle fiber during the cell division. When you will study about the cell division, you will come to know about it. After the completion of this cell division, these two chromatids are known as the chromosomes. Now, a typical chromatin material consists of DNA, that is deoxyribonucleic acid, and histone, which is a type of protein. The DNA is about 40% and histone is about 60% in the chromatin. Here is the structure of the highly condensed chromosomes, and it is unwinded. You can see nucleosomes, DNA, everything, chromatin fiber, chromosome, everything is visible. This you will find it in your book. We are now going to study about the DNA. The DNA stands wind around a core of 8 histones. Means it contains 8 histones. One DNA core contains 8 histones. You can see histones, which are known as nucleosomes. And this repeating components of each DNA strand lengthwise is known as the nucleotides. When I will send you the notes, you will get all these things. Now we are going to study about the structure of DNA. The shape of DNA molecule was studied first by Rosalind Franklin in 1953, and its work was done by Watson and Crick in 1953 itself. The double helical structure of DNA. This is a double helical structure of DNA which was given by Watson and Crick, for which they got the Nobel Prize in 1953. DNA is a very large molecule and it is described as a macromolecule. It is composed of two complementary strands. You can see over here two strands. These are the two strands. Two complementary strands. Two are complementing each other, and they are wound around each structure. They are wound around each other, and it's called as double helix. You can see here one complete turn. I have marked here one complete turn. You can see it from the book itself. And you can see in between them I have marked with different sketch colors, and also I have written there CGTA. You can see it everywhere. This forms a ladder like structure. This C stands for cytosine, G stands for guanine, A stands for adenine, and T stands for thymine, which are known as the nitrogenous base. They are binded with each other with the hydrogen bonds. In the chemistry also you have studied about the triple bond and the double bonds. These bonds are known as the hydrogen bonds. The DNA consists of, the nucleotides of the DNA are made up of 3 components. That is, first one is phosphate, second one is sugar, and third one is base, that is also known as the nitrogenous base. These are attached just like this. You can refer to your book. If it is not clear, refer to your book. So they are just attached with each other this way. You can see 2 bonds. These are the hydrogenous bond. This is the phosphate. This is the phosphate sugar, and this is the phosphate, and this is the sugar. So typically this is the structure of one nucleotide. It consists of 3 components. Formation of new DNA is not there in your syllabus, so we are not discussing about it. We'll move to the next topic, which is the last topic, that is the genes. Genes are highly specific sequences of nucleotides. We have seen here the nucleotides on a chromosome that encode a particular protein which express in the form of particular feature of the body. Your particular feature of the body is encoded inside the genes. The question answer and all the other notes will be forwarded to you soon. So I am winding up. Hope you understood this one. Thank you.